Mrs.G.NITHYA, Assistant Professor, Department of Biochemistry, Deakin College for Women, Vellore. Today I am going to take innate immunity under the topic types of immunity. So first we should know what is immunology. So immunology is the study of immune system in all organisms. It deals with the physiological functioning of the immune system in state of both health and diseases. Okay, then what is immunity? Immunity is the body's ability to fight off harmful microorganisms such as fungi, protozoa, bacteria and viruses. Okay. Then what is immune system? So immune system will produce the immune response in our body. Okay. So it produces two major steps. That is, one is recognition. It can produce the immune response by means of two major steps. That is, one is recognition and another one is response. After proper recognition only, we can produce the immune response. And immune system comprises immune organs, that is lymphoid organs, that is primary lymphoid organ and secondary lymphoid organ, and immune cells. Regarding immune cells, that immune cells can be synthesized from pluripotent stem cells by means of hematopoiesis. Okay, so that pluripotent stem cell hematopoietic reaction can be divided into two lineages, lymphoid lineage and myeloid lineage. Lymphoid lineage synthesize A granulocytes such as B lymphocytes and T lymphocytes, and myeloid lineage synthesize granulocytes such as neutrophil, macrophages and dendritic cells. Then coming to the types of immunity. The immunity can be broadly classified into two major types. One is innate immunity and another one is adaptive immunity. So what is innate immunity? It is a non-specific immunity. Our body pre-existing arrangements can produce the innate immunity. Okay. Then what is adaptive immunity? It is a very specific immunity. Okay. The immunity produced in this adaptive immunity is very specific in nature. Okay, here it is otherwise known as acquired immunity. Why it is known as acquired immunity means during our lifetime we are facing so many types of pathogenic microorganisms in our daily life. So for that pathogenic microorganisms we are producing immune response. So we are acquiring that type of immunity during our lifetime. It is otherwise known as acquired immunity. Adaptive immunity is otherwise known as acquired immunity. So host defenses can be classified into this type of classification. That is, it is a type innate immunity. It is again classified into first line of defense and second line of defense. Under the first line of defense, we have so many barrier systems. That is physical barriers, chemical barriers. And in second line of defense, we have inflammatory barriers. Then various insoluble factors such as interferons and phagocytic cells and complement. If the first line of defense, that is a pathogenic microorganism, it evades the first line of defense, then it can go for second line of defense. Again, if the pathogenic microorganisms evade the innate non-specific immunity, then it can go for acquired specific immunity. See, if a body's immune system protects from pathogenic organisms with increasing specificity, most simply, physical barriers prevent pathogens such as bacteria and viruses from entering the body. If a pathogen breaches these barriers, then the innate immune system can come and give its role. Okay. If pathogens successfully evade the innate response, the vertebrates possess a third layer of protection called adaptive immune system. So we have a deep discussion about the innate immunity. So innate immunity comprises four types of barrier system. One is anatomical barriers, then second thing is phagocytic barriers, then inflammatory, then physiological barrier system. So in that already we have seen that in innate immunity itself we have first line of defense and second line of defense. Under the first line of defense we have anatomical barriers. So the barrier system given here is anatomical barriers. In that we have skin, that is epidermis and dermis part of the skin, and mucous membrane covering of respiratory tract, gastrointestinal tract and genitourinary tract. Then lacrimal apparatus, which can synthesize tears and saliva, and various chemicals present in that anatomical barriers are given here. So it is a diagrammatic view of skin, that is it expresses various layers, that is epidermis, dermis and subcutaneous tissue, and in that it contains sebaceous gland. Okay, so that sebaceous gland secretes fatty acids and lactic acid. That fatty acids and lactic acids will be very helpful for the maintaining of pH, that is acidic pH in the skin, that is 4 to 6. So it can prevent the entry of the pathogens. So first line of defense, it explains that is it will be very helpful at the entry site of the microorganisms. It prevents the entry of the pathogen itself, so that it can act as a mechanical and chemical barrier in this. Then coming to mucosal membrane. So mucosal membrane of respiratory tract contains hair-like projections called cilia. So that cilia, it is a hair-like projections. It provides a synchronous movement. It propels away the microorganisms by producing the synchronous movement. Okay, so it propels away the mucus entrapped microorganisms. And it also contains lysozyme, mucopeptide and secretory IgA, which has a microbicidal activity. And lacrimal apparatus, it is a physiological system present in our eye, and it can synthesize tears. Okay, and it also contains drainage. Okay. Then the lacrimal apparatus, it secretes tears. That tears mechanically remove the foreign particles, and also it contains lysozyme, which is a hydrolytic enzyme. Okay, so that hydrolytic enzyme, it breaks the glycosidic bond between the NAG and NAM. What is NAG? N-acetyl glucosamine and N-acetyl muramic acid. So which is the backbone of peptidoglycan layer of bacteria. Okay, so that lysozyme will break up that backbone of that bacteria, so that it can causes the lysis of that bacteria. In saliva. So that saliva, it is also considered to be a first line of defense. It mechanically washes away the pathogens from your teeth, and it reduces the number of pathogens, and it is also contain secretory IgA. Okay, so that secretory IgA which coats and protects our teeth. Okay, so from harmful bacteria, it protects our teeth from our harmful bacteria that may cause tooth decay. Okay, so it secretes secretory, that is it secretes IgA, that is secretory IgA. That secretory IgA which coats and protects every tooth from harmful bacteria that may cause the tooth decay. Then coming to second line of defense. So here here it is a phagocytic cell, and it shows the phagocytic reaction. So the second line of defense can also bring out by the phagocytic cells. Phagocytosis is known as that is engulfment of foreign particles. That phagocytic reactions can be mediated by various phagocytic cells such as macrophages, neutrophils, eosinophils and all. Okay, so here the phagocytic cells, at first bacterium becomes attached to the membrane and it forms a pseudopodia. So it first attached with the macrophages cell membrane, and that membrane forms pseudopodia. Okay, so then the bacterium is getting ingested into that cell so that it can form phagosome. Then it can it can fuse with the lysosome so that it can form the phagolysosome. Then that lysosome releases lysozyme and it digests that bacteria, and the digested products can be released, can be exhaled from the phagocytic cells. So this is known as phagocytic reaction. So here it shows the engulfment of foreign particles by phagocytic cells. Then third thing is inflammatory barriers. So the inflammatory barriers produce inflammatory response. Okay, so that inflammatory response can be bring out by means of various vasoactive amines. Okay, that is known as chemotactic factors. So the tissue damaged by a wound or by an invading pathogen can produce the inflammatory response. So inflammatory response is due to the release of chemical mediators from tissue damaged lymphocytes and invading microbes itself. Okay. So what are the chemical mediators? The chemical mediators are histamine, fibrin, kinin and cytokines. So the release of the chemical mediators can cause the vasodilation of capillaries. What is vasodilation means? Increase in the diameter of blood vessels. So that is increase in the diameter of blood vessels. It causes redness of tissue and there is increase in the tissue temperature and increase in the capillary permeability. So due to the increase in the capillary permeability, there is more influx of fluids and phagocytic cells from the bloodstream into the tissue site, thereby into the infection site. So the inflammation causes redness, heat. So due to the capillary dilation and increased blood flow, then it causes swelling. So as I already told that there is an influx of fluid from bloodstream into the tissue site. No? So it causes swelling, that is edema, and pain. So due to the tissue destruction, it causes pain. So this diagram shows the inflammatory response. So the inflammatory response is a series of cascade events. Okay. So if there is any tissue damage, okay, by a wound or by invading pathogen, so the tissue damage causes release of vasoactive amine and chemotactic factors. So thereby it produces, that is it causes increased capillary permeability. So that increased capillary permeability causes movement of fluid and immune cells from the bloodstream into the tissue site. Okay. Then so that movement of immune cells, it causes margination, extravasation. So what is margination? Attachment of immune cells along the epithelial cell membrane. And what is extravasation? So it acts as a transmembrane compound in between the epithelial cells, so that it is known as extravasation or diaperesis. Okay. Then finally that phagocytic cells enter into the tissue part and thereby into the infection part. Okay. So the accumulation of phagocytic cells into the tissue damaged site is known as chemotaxis. That chemotaxis can be bring out by means of chemotactic factors, that is vasoactive amines. So finally the bacterium will be lysed. Then what are all the chemotactic factors, that is vasoactive amines that can be released, and what are the role of that means? Histamine and kinin, it causes vasodilation, increased capillary permeability of blood vessels, and prostaglandins are needed for intensifying the action of histamine and kinins. Then leukotrienes are needed for increased capillary permeability and phagocytic attachment. Then finally physiological barriers. So the physiological barriers that contribute to innate immunity include pH, temperature and various soluble and cell associated molecules. So in that pH and temperature, that is regarding pH, gastric acidity. So in our stomach, the usual environment maintained in our stomach is acidic, is it? So that gastric acidity is an innate physiological barrier to infection. Only few ingested microorganisms can survive that acidic pH, that low pH of the stomach contents. And some species normal body temperature itself, it inhibits the growth of the bacteria. For example, chickens have innate immunity to anthrax because of its high body temperature. Okay. So in this way, pH and temperature contribute to the, that is, these are the factors which can contribute to the innate physiological barrier. Then complement system. It is an important system, and this in this complement system we have, that is in our blood we have 20 different proteins. Okay, these can also produce a cascade reaction. So that is known as complementary mediatory reaction, that is classical pathway and alternate pathway of complement mechanism. Okay, so that complement proteins are present in inactive state only. So after it gets proper stimulation, it gets activated and it mediates various complement mechanisms. Okay, for example, see how it can lyse that bacterium or any other pathogenic cells means complement proteins insert themselves into the membrane of pathogens creating a pore. So the complement compounds, it attach with the pathogens and in surface it will create a pore. Okay, so through that pore various hydrogen and ionic particles enter into that pores so that the cell becomes swells and finally it gets lysed. Then finally interferons. So the interferons are proteins present in the body. Most probably we call it as cytokines. It is a type of cytokines only. Okay. So that interferons roles is very important, particularly in viral infection. So once if a cell is infected by a virus means that infected cell will produce and secrete cytokines. So that is interferons. So that interferons will produce the generalized antiviral state to the nearby cells. Okay. So it produces the antiviral immunity in a generalized manner. See. If the cell is infected by a virus means it activates, that is it causes transcriptional activation of interferon genes. Okay, so that interferon genes, it undergoes transcription, that is translation, and it will synthesize a protein called interferons. So there is a synthesis of interferons will occur. After secretion of that interferons, that secreted interferons will go on bind with the nearby cell which has a receptor for that. Okay, so it can go and bind with the interferons receptor on that membrane of that cell, and finally it causes the transcriptional activation of antiviral genes. Okay, so that the antiviral protein can be synthesized. So in this way it produces antiviral protein, thereby it creates the antiviral immunity. So the secreted interferons will go and bind with the receptor present on the membrane of the nearby cell, and it produces the antiviral protein so that it will produce the generalized antiviral immunity in the nearby cell. Okay. Okay, so in today's class we have discussed about the type of immunity such as innate immunity. So in that 4 important barrier systems play an important role. So what are that means? Anatomical barriers, phagocytic barriers, inflammatory barriers and physiological barriers. Okay, thank you.